So, welcome to this content review looking at the topic of homeostasis and doing a little bit of revision on some of the key concepts. So, for this, what you'll need is you'll just need a pen and paper and obviously just some space to work in. And what we're just going to do is just go over some of the key concepts that are presented to you in the unit on homeostasis that you'd probably studied in year 10. So, on your paper, what I'd like you to do just at the top is just pop yourself a nice title of homeostasis, okay? So what we're just going to do is just going to build a couple of key examples here in a set of notes that you can then use just for your own revision, and obviously using it to help you just apply it to different specific questions that we might be asking you to have a go at in coming days. So, as it says, homeostasis at the top. Now, homeostasis, we need a key definition to begin with. And that key definition is that homeostasis is the control of internal conditions of a cell or organism. So it's control of the internal conditions of a cell or an organism in order to maintain optimum conditions. Now, that can seem slightly complex, so what we're just going to try and do is try and break that down a little bit. So, homeostasis is obviously firstly something that is carried out in living organisms. Process that's carried out in living organisms. Okay, now in terms of what happens, it's the control of the conditions within an organism, whether that's cell, whether that's a larger organism, in order to maintain optimum conditions. What we mean by optimum conditions is order to keep that organism So it's to keep the organism functioning at its best. So the internal conditions inside the organism that allow it to function at its best, okay? And these control mechanisms that are used are often carried out. So homeostasis are often carried out in response to internal or external changes to the organism or its surroundings. Now, what are some of the things that can be controlled within an organism in order to try and maintain its best optimum internal conditions? Now, what homeostasis is often trying to do, it's often trying to maintain conditions that allow enzymes to work. So remember when we've drawn enzymes, got that active site for bonding to other molecules and either building them or breaking down molecules in metabolic reactions. So, what are some of the conditions that the body tries to maintain and control in order to allow these enzymes to function as best as they possibly can? So, some of the things that it controls are body temperature. You enjoy yourself a little monitor just to represent that. So it controls body temperature. What else does it control? Controls blood glucose levels or blood glucose concentration. Blood glucose concentration. Might have a red pen, but there's some blood droplets. And a nice little sugar cube, just to represent both of those. And then the other thing that it helps to control within the body as well, homeostasis, it's the body or organisms trying to control water levels, which is often referred to as osmoregulation. Now, it's important to bear in mind that the maintaining of these internal bodily conditions means that the body has to act to change body temperature, blood glucose concentration, water levels, and things like that. Now, a lot of these responses are what you might describe as automatic. So they're done subconsciously, they don't require thought. And the maintaining of these often requires the action of effectors, which are either glands or muscles. And particularly when we're talking about glands are incredibly important because they release hormones, which are often responsible for correcting levels back to their optimum. So, a little overview there of what homeostasis is. Now, we're going to have a look at an example of how homeostasis occurs, looking particularly at blood glucose concentration and how it's regulated using hormones via a process that you might see referred to as the negative feedback cycle. So, you've got your notes there, you can pause your video wherever you need to, and then if you just get yourself a second page, we're just going to go over negative feedback and what that looks like in regulation of blood glucose concentration. So, first and foremost, what we are looking at when we're talking about the negative feedback cycle is we're talking about, got out of focus, there we go, come back into focus. We're talking about the restoration to an optimum level. Restoration to an optimum level. So in the case of our example, we're going to be talking about blood glucose concentration. We're talking about blood glucose concentration. Okay, so what we're talking about is if we start with this idea of blood. Now, our blood can sometimes contain too much glucose. So I'm just going to draw lots of G's with lots of G's, and these can represent our glucose molecules. So these represent our glucose molecules, okay? Now, what happens when the blood glucose concentration gets too high is that the body wants to try and lower it so it's back to an optimum level. Now, that's where the pancreas comes involved. Now, the pancreas is an effector because what it does is when the body detects that there's too much glucose in blood, it causes the pancreas to release lots of a specific hormone, insulin. So insulin's released. Now, as we say, the body receptors detect high levels. Now, once they've done that, that insulin is then released. And insulin is a hormone. It's a hormone. Now, that hormone then goes and acts on the glucose. And at the liver, it's a bit of a weird island-shaped organ. Just sort of draw it like that. So, in the liver, the glucose is then acted upon by the insulin, and it's bonded together and stored. So, the glucose is stored in the liver as glycogen, which then means that the glucose concentration in the blood has decreased. Now, it can work the other way as well. So if the body's blood glucose concentration gets too low, then what happens is that the body then detects this again, but instead what we have is we have a different hormone that acts on the glycogen in the liver. So if we just draw a second liver, so remember up here we're talking about high blood glucose concentration. Here we're talking about if the blood glucose concentration is too low. So if the blood glucose concentration is too low, what the body's wanting to try and do is increase the concentration of blood glucose. So it has all this glucose that has previously been stored up as glycogen molecules in the liver. So if we just draw that out again, as you'll see, 2, 3, 4, 5. So this is our glycogen. Now, again, the same similar process happens as in receptors detect low levels. So low levels of glucose. So in response to that, then causes the pancreas to release a different hormone. And what it causes it to do is it causes it to release, just do a capital G here to show the difference, a hormone called glucagon. So glucagon is released, which again is a hormone. And that glucagon causes the glycogen to break apart back into glucose. So the blood then becomes full again of glucose molecules, and the blood glucose concentration increases. So, blood glucose concentration decreases, whereas the other way, because the glucagon acting on the glycogen, the blood glucose concentration increases. So there you have an example of a negative feedback cycle in which the body is acting to restore, drop that back up, restore levels of particular things like blood glucose concentration back to their optimum level through receptors detecting change, and effectors such as muscles or glands like the pancreas releasing hormones responding. So you've got a full set of notes. Pause the video, make your own notes as you can, and then you can use some of these notes to have a go at some of the particular questions that we might set you as a result, either in remote work or further on in normal classwork. So thank you for watching this summary of homeostasis and examples of negative feedback cycle.